Well, hey, good morning, everybody, and happy snow day. Hope you're doing well. So, today we're going to jump into a new chapter, chapter 18, looking at how we control the expression of genes. You understand sort of the mechanisms of transcribing, translating the information from DNA into a polypeptide sequence that'll fold up and become a protein. Now, in chapter 18, we look at how do we actually manipulate or control that. So, I'm using a presentation originally developed by David Konefsky, another AP bio teacher, and sort of taking parts of that to see what we need here. So the big question is, how do we control the production of proteins? So we're going to look at this in two different segments. One is looking at what prokaryotes do, and the other is eukaryotes. So let's begin with prokaryotes. And we know that there is this direct connection between transcription and translation in prokaryotes, and that's because they don't have a nucleus. Since they don't have a nucleus, as soon as transcription begins and you make that messenger RNA, there's no editing or anything like that that goes on. Instead, what you do is immediately begin translation. So basically, if you start transcribing, you're going to start translating. So what that means is if you're going to control genes, if you're going to be able to switch genes on or off, we need to do that through controlling transcription. We need to do that through controlling access to the DNA. So here we see, again, just sort of that connection between transcription and translation. The DNA is in blue, the messenger RNA is in red, and then the polypeptide's in purple there. So as soon as RNA polymerase zips along and starts transcribing DNA into messenger RNA, then the ribosomes attached to the messenger RNA begin translating it into the polypeptide sequences. So again, what we need to do is basically prevent this transcription from happening. Now, a lot of times we know this happens through negative feedback, that some molecule that's produced will actually inhibit its own production. Okay, so here we see the different enzymes that are created, and the idea is some enzyme in the process will actually inhibit its own production, and we'll see ways in which that happens here shortly. Now, the sort of structural components that help regulate gene expression in prokaryotes are called operons. So an operon is a section of DNA and then the molecules that help sort of regulate that. Okay, so again, it's sort of a cluster of a gene, which is instructions for making certain proteins. The promoters, which we know the promoter is where the RNA polymerase docks. And then there's this new structure called an operator. An operator is a section of DNA that basically acts like an on-off switch. So that's what I always think of operators as. So again, think of an operator as an on-off switch. And all these parts work together. Now, there are two types of operons. There are inducible and repressible operons. To induce means to force something to happen. So inducible operons, they're normally switched off, but they can be forced on. Repressible operons, to repress is to hold back or hold down, right? So repressible operons are normally on, but can be forced off. So it's basically ways of switching gene expression off or on. Here's just sort of a schematic of an operon. You have the promoter. Again, that's where RNA polymerase attaches. You have these structural genes that will be used to make whatever enzymes you're looking for. And again, that operator is your on-off switch. Upstream of this, or before this, is a regulatory gene. We'll talk about its relevance here shortly. Okay, so let's begin by looking at inducible operons. Again, inducible operons, they're normally off, but they can be induced or forced on or switched on. And the example they use in the AP curriculum is the lac operon. The lac operon has genes that are used to produce the enzymes that will ultimately create lactase, the enzyme that breaks down lactose. Now, lactose is milk sugar, and this is in E. coli. Now, E. coli, they don't make their own milk, so they're generally not needing to produce this lactase enzyme, right? The only time E. coli would need to produce the enzyme to break down lactose is basically when, like, we or some other mammal feeds the bacteria lactose. So if you consume dairy products, you consume lactose, that goes into your guts, and then the E. coli in your guts are basically fed some lactose. So normally E. coli have this gene switched off, or this operon switched off, but it's switched on when they're given food. So how does that happen? Well, let's look at the parts here again. Again, the promoter, that's the region on the DNA where RNA polymerase will attach. And then you have these structural genes here. They mention like LAC-Z. So those are the genes that produce the enzymes to break down lactose. But then remember that off-on switch? That's the operator. Now what happens is a molecule called a repressor docks on the operator. So when the repressor docks on the operator, look what it does to RNA polymerase. It basically blocks RNA polymerase. And if RNA polymerase gets blocked, then it won't transcribe the genes, and basically the operon is switched off. Earlier, we talked about the regulatory gene upstream of the operon. What the regulatory gene does is it provides instructions for making the repressor. Okay, so the regulatory gene codes for this repressor protein. The repressor can dock on the operator, and when it does, it prevents RNA polymerase from attaching. When RNA polymerase doesn't attach, you don't transcribe the genes, and you don't make the proteins. Okay, so again, this would be sort of the normal state for this lac operon, because typically E. coli don't need to be making the enzymes to break down lactose. Again, it's only when food is present. All right, so again, lactose, this is sort of the standard situation. Lactose is absent. Repressor docks in the operator. That blocks the RNA polymerase. RNA polymerase can't transcribe those structural genes, the genes that are used to make the enzymes that you make lactase with, and the operon is switched off. Now, what happens if you have some dairy, or you have some ice cream or some milk, and you send lactose into your guts? Well, a version of lactose, it's called allolactose. This is an isomer of lactose. Look at what it does. It docks on the repressor, and then shape and function. Repressor changes its shape. If this repressor changes its shape, is it going to be able to dock on the operator anymore? Well, no. That allows RNA polymerase to attach. It transcribes those genes. You make these enzymes that are used to make lactase, and then lactose gets broken down. Well, think about what happens. When lactose gets broken down, then the allolactose will be gone. The repressor will go back to being active, and then it docks on the operator, and you switch transcription off again. Okay. So when lactose is present, the molecule allolactose, or version of lactose, they call it an inducer. It docks on the repressor, changes the shape of the repressor. Repressor can't dock on the operator anymore. So then RNA polymerase get attached to the promoter and transcribe those genes. Once you transcribe the genes, you make the enzymes that will ultimately make lactase. You break down lactose, and then the repressor goes back to its active conformation and docks on the operator and stops production. So again, some end product, something you create like lactase, will actually inhibit its own production. All right, because it will break down the lactose and then switch the operator back on, or the repressor back on. So good example of negative feedback, right? You only make what you need, you break it down, then you stop making it. Okay, now the opposite of this is a repressible operon. Repressible operons are normally switched on, but can be repressed or forced off. An example of this is the TERP operon. The TERP operon is significant because it's used to produce tryptophan. Tryptophan is an amino acid. Okay, so you need amino acids to make proteins, so it makes sense that bacteria would produce tryptophan if they can. But what if you have a big tryptophan-laden meal? You eat a bunch of Thanksgiving turkey. Well, if you're giving the bacteria a bunch of tryptophan, why make it? So in the case of feeding bacteria tryptophan, then they'll switch off the TERP operon. So in this instance, normally the repressor that's created is switched off. It's an inactive conformation. If the repressor is normally switched off, well, then the operator is open for business here. RNA polymerase zips along. You make these molecules that can then join together and make up the enzymes that will produce tryptophan. Okay. So again, the standard situation here for the terp operon is that it's normally on. So the repressor, when it's created, is normally inactive. RNA polymerase transcribes the genes, and then you make these enzymes that'll make tryptophan. So again, this is normally switched on because you normally have to make tryptophan to incorporate into proteins that the bacterial cell needs. But again, you have a big meal that has a bunch of tryptophan in it. Here's the tryptophan. Well, look at what the tryptophan does. Docks on the repressor. The repressor switches to an active conformation or an active shape. It docks on the operator, the on-off switch, and then transcription of those genes stops, and you stop producing tryptophan. So the bacteria stop making tryptophan. They'll incorporate the tryptophan amino acids that they've been given into different polypeptides or proteins. And then tryptophan levels in the bacterial cell begin to drop. When tryptophan levels in the cell drop, then the repressor goes back to being inactive and the bacterial cell starts making its own tryptophan again. So a really cool way to regulate tryptophan production. Again, a good example of negative feedback. All right, now, these switches aren't just on-off switches. They're also like dimmer switches. So you can raise production and lower production. And this is sometimes referred to as upregulation or downregulation. So now we're going to look at how do you upregulate or downregulate the production of enzymes for the lac operon. So if you recall from the last chapter, RNA polymerase doesn't just sit on the DNA. It has to dock on some transcription factors that sort of act like car seats for the enzymes. And here, the transcription factor is called CAP. Okay. Now, normally CAP is in an inactive shape. It's not going to interact well with RNA polymerase, and that sort of prevents RNA polymerase from doing very much. But there's this relationship between levels of glucose and CAMP, a second messenger in cells. So it's sort of cool because the question is, if you give a bacterial cell lactose or glucose, what's it going to burn first? And you can imagine if all life has the enzymes for breaking down glucose, then glucose is going to be the preferred fuel. So here's the relationship. When glucose levels are low, CAMP levels are high. And when glucose levels are high, CAMP levels are low. So there's an inverse relationship between glucose and CAMP. So if you have lactose and you have low levels of glucose, then look at what happens. Remember, if glucose levels are low, CAMP levels are high, CAMP docks on CAP, CAP becomes active, and RNA polymerase gets situated, you transcribe the genes. So again, if you have lactose and low levels of glucose, you basically situate the RNA polymerase so it can transcribe the genes, and you break down the lactose. You use lactose as fuel. But what if you have lactose, but you also have high levels of glucose? Remember, if you have high levels of glucose, you have low levels of CAMP. If you have low levels of CAMP, then CAP stays inactive. And so even though the repressor has been activated, if CAP is inactive, RNA polymerase can't dock very well, and transcription of the lactase genes are pretty low. So what that means is the cell will first burn through the glucose and use that up, and then when glucose levels are low, then it will start to break down lactose. So again, it's not just an on-off switch. It's almost like a dimmer switch. You can brighten things or dim things, or you can upregulate or downregulate. So pretty cool examples. So that's prokaryotes. We'll pause here, and then we'll get into eukaryotes.